Hello, everybody, and welcome to today's webinar, Unlocking the Proteomic Landscape of CSF-Derived EVs, a new pathway for neurology biomarker discovery. I am Sydney Panilla of LabRoots, and I will be your moderator for today's event. Today's web seminar is presented by LabRoots and brought to you by Olink Proteomics. To learn more, visit olink.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues you have here as well, if you happen to have any trouble seeing or hearing the presentation. Okay, I would now like to welcome our Olink moderator for today's event, Ryan Lammers, PhD, Product Management, Olink, part of Thermo Fisher Scientific. Ryan, I'll go ahead and turn it over to you.
Okay, great. Thank you so much. And thank you all for joining us today on our webinar, Unlocking the Proteomic Landscape of CSF-Derived EVs. So to get started today, I'll give just a very brief intro to Olink Proteomics and PEA technology, just to make sure that everyone's up to speed and has the same background required for our feature talk today, which will be delivered by Professor David Walt. Following Dr. Walt's presentation, we'll have some additional topics in EV research. I have just a couple additional case studies highlighting the use of Olink in EV research, followed by a brief Q&A session with what time we have remaining. So as a brief background to Olink, Olink is a proteomics company, which is now part of Thermo Fisher Scientific. And since our commercial launch in 2016, our mission has been to expand the coverage of the human proteome. And with that in mind, we've had a continuous drive for innovation. As you can see from this timeline shown on the screen, we have a track record of continually launching new products with new features to advance proteomic research by, for example, increasing throughput, increasing complexity, and of course, offering customizability in the proteins that can be targeted for each specific research project if desired. In the bottom, as is shown with this line graph, the technology has been well accepted throughout the research community, where we now have greater than 3,000 publications leveraging Olink across many different therapeutic and research areas. And at the core of our technology is PEA, which stands for Proximity Extension Assay. And what this means is that for each analyte that we target, 2 antibodies must bind. And appended to each of those antibodies are complementary single-stranded oligonucleotides that are specific to each analyte of interest or being targeted. When the 2 antibodies bind to their correct analyte in proximity to one another, the oligos hybridize. And then through multiple rounds of PCR amplification, the signal is then amplified and read out using either qPCR or next-gen sequencing, depending on the product that's being used and the complexity of that product line. So this multi-factor authentication is what drives our high level of specificity, requiring both the dual antibody recognition and the oligo hybridization ensures that you are testing the protein that you think you are actually measuring. And then subsequent to that, leveraging PCR then affords us the extreme sensitivity that we have become known for. And because of the high sensitivity of PEA, we derive a substantial benefit in helping researchers access the low abundance proteome, which is often inaccessible with other technologies. And as I'm showing here in this graph, the bottom right is the graph overall that is a descending order of proteins down to lowest abundance in the bottom right. And I just wanted to highlight that the bulk of our products target these low abundance proteins, such as chemokines and cytokines and things of that nature. And over the years, we've seen a substantial benefit to identifying additional protein biomarkers or biomarker signatures for diseases that were commonly or previously elusive, such as ovarian cancer detection, identifying novel potential immunotherapeutic targets, for example, in melanoma, and identifying protein biomarker signatures now for COVID severity. Analyzing protein biomarkers becomes notably challenging when working with certain sample types. For example, if they're valuable or rare or collected using invasive methods. Or for example, when samples are processed with challenging workflows, as we'll hear about today, such as microvesicles or extracellular vesicles. However, PEA enables actionable insights from as little as 1 to 2 microliters of those precious samples, which, as we will hear today, is valuable when working with such sample matrices as EVs or CSF. And so with that brief background, it's my privilege to introduce to you all today Dr. David Walt, who will deliver our featured talk. Dr. Walt is a professor of bio-inspired engineering and professor of pathology at Harvard Medical School and Brigham and Women's Hospital, where he works on developing new diagnostic tools and new biomarker assay technologies to address unmet needs in clinical diagnostics. His lab applies such new technologies to the early detection and diagnostics of diseases and disorders of heightened clinical importance, such as cancers and neurodegeneration. Dr. Walt is the founder and co-founder of multiple known life sciences and biotech companies, which I'm sure you have all heard of. And among his many accolades, Dr. Walt is a fellow of the National Academy of Inventors, and several years ago was inducted into the U.S. National Inventors Hall of Fame. Most recently, in 2025, he also received the National Medal of Technology and Innovation from President Biden. And so with that, Dr. Walt will discuss some of his recent studies aimed at uncovering biomarkers in brain-derived EVs.
Thank you, Ryan. So before I start, I'd like to put the audience at ease. 2 weeks ago, I had a minor procedure on my vocal cords, so my voice sounds pretty rough, but it doesn't hurt, and I'm prepared to give this talk, and we'll see how it goes. But it doesn't hurt, so just wanted to make sure everybody was comfortable. So today I'm going to tell you how we used an Olink panel to help us identify candidate markers for EVs. First, I'm going to provide you with some background on EVs, as well as describing our initial attempts to identify brain-derived EVs that where we ran into some serious issues that led us to use the Olink technology to help us solve the problem. So before I get into the presentation, I have to, as a requirement of my position as a professor at Harvard Medical School, disclose my conflicts. The two that are a bit relevant today are Quanterix, where I'll describe how we used our Simoa technology that originated from my laboratory to solve some of the problems. And I'm on the board of directors of Quanterix and have equity in the company. And also Everest Biolabs, which was recently founded to do high throughput EV purification. And so those are really the two conflicts that are relevant today. So this slide represents how we presently learn about the brain. We currently don't have methods to access the biochemistry of living patients as they develop, as they age, as they acquire brain insults, for example, through head injuries, and how they respond to things like medication. The vast majority of what we know from a biochemical perspective comes from the genetic code that a person starts with. That is their predisposition based on a few genetic variants. And also, we can further understand a little bit about those individuals by analyzing their brain tissue post-mortem. So that's obviously something that doesn't apply to a living patient. So gaining access to the underlying biochemistry would fundamentally change how we understand the human brain, how we can use that information to help us develop drugs, to diagnose and monitor brain-based diseases like neurodegenerative diseases, psychiatric diseases, and so on. So ideally, we want to be able to take different cell types from the brain and measure those changes in protein concentrations, in RNA expression, both before and after the development of neurologic and psychiatric diseases, and also before and after treatments are administered. That's how we'd optimally like to learn about the brain. But clearly, under present circumstances, we can't just take brain tissue because living patients are unwilling to give us pieces of their brain to study. And so the question is, what's the next best thing to actually obtaining brain tissue? So our idea is to use something called extracellular vesicles. These are more commonly called EVs, and I'll refer to them as EVs the rest of the talk. And we want to use these as a window into the brain. EVs are nanometer scale vesicles that bud from all cell types. They contain, as you can see in the slide, proteins, RNA, lipids, and other molecules that regulate cell function. They circulate in biofluids such as plasma, cerebrospinal fluid, also called CSF. They also have been found in urine, in saliva, and also recently were used and isolated from tears, from which they all can be isolated to serve as what we call molecular snapshots of their cell of origin. So as you can see on the right side of the slide, there is the RNA in the EVs, at least EVs in cell culture, have a very high correlation to what's present in the parent cell. And this is just a correlation, as you can see, between the cell, which is on the x-axis, and the EVs from those cells on the y-axis. So we can think of this as a snapshot, although there's evidence that there might be some specificity with respect to the types of RNA molecules and the types of protein molecules that actually get packaged before they're released from the cell. So even though in this particular case you can see that there's good correlation, there are some cases where specific proteins are either more, and specific RNA sequences are more or less specifically incorporated into EVs. And those might actually be even more useful, for example, for performing a liquid biopsy. So a major goal has been to specifically capture EVs from particular tissues and organs. And this is accomplished typically by using antibodies that specifically pull down cell type specific EVs. My lab studies multiple diseases, including multiple cancers, multiple neurodegenerative diseases, some psychiatric diseases, and also infectious diseases. For our neurological work, we have been trying to identify markers that will enable us to isolate brain-derived EVs. And the best place for us to start is CSF. But of course, our ultimate goal is to isolate brain-derived EVs from blood plasma, because it's obviously much easier to collect. Cursor keeps moving, so it's important to understand that there are many publications that claim to measure brain-derived EVs. If you go search the literature, you'll find hundreds and hundreds of publications that claim to use brain-derived EVs. Unfortunately, and that's one of the reasons I'm talking to you today, they do not actually report on brain-derived EVs. There have been huge methodological challenges in the field. First and foremost, as we showed in our 2021 Nature Methods paper, there's a particular marker called L1-CAM that is the most commonly used neuron target, which we found unequivocally is not associated with EVs. And so if you read these papers on brain-derived EVs and you look, you'll see many researchers are using L1-CAM to pull down those, what they call brain-derived EVs, from a particular biological fluid like CSF or blood, and then they analyze that. What I'll show you from our Nature Methods paper, what I'm showing you from our Nature Methods paper here is that what you can see on the top is that the tetraspanins, so these are three transmembrane proteins, CD9, CD81, and CD63. This is a size exclusion chromatography fractions, and so I want to make sure everybody can understand this as we go through the talk today, is that the early fractions in size exclusion chromatography have the larger molecules or the larger components, and that's where the EVs are because they're nanometer sized. The free proteins actually come in later fractions, and so you can see that when we analyze the EVs that are coming out of the size exclusion chromatography column, the CD9, CD63, CD81 all correlate with the presence of those early fractions, which correlate with the EVs. You now see on the bottom side, on the bottom screen, albumin, which is on the bottom left. You see that albumin comes in a later fraction. And so when we actually run an SEC and we run L1-CAM assay using the ultrasensitive method called Samoa that I referred to before, you see that L1-CAM actually elutes in the free protein fraction. What this means is that L1-CAM is not associated with the EVs. And we showed this unequivocally in our Nature Methods paper in 2021. And so, again, now we have to find something that is specific to brain-derived EVs. The problem, however, is not only with the transmembrane proteins that are secreted, or in the case of L1-CAM, it turns out that the L1-CAM that everybody's using to pull these EVs down turns out to be either a splice variant that does not contain the transmembrane protein, or it's a protein that actually gets cleaved. And so that's why it's not associated with EVs. But many proteins turn out to be non-specifically bound to the EV, or they have the wrong topology. And this can best be addressed using what we've developed, which is called a proteinase protection assay, which chemically removes all the proteins from the outside of the EV while preserving the contents of the EV. So the EV, because it's a membrane protected, it's a membrane that encloses the cell contents. We can use this protein protection assay with a protease to just chew up everything on the outside of the EV and only retain the internal condition of the EV. And so the proteins on the outside, after this assay is performed, will show nothing in the internal condition, while the proteins that are internal should have 100% recovery in the internal condition compared to the no treatment control. And so by conducting this extra layer of validation, we were able to show, for example, that a protein called CMP, a target that was published in literature to capture a subset of the brain-derived EVs, which putatively was able to pull down oligodendrocyte specific EVs, actually turned out to be internal. And so you can see this is the non-treated, and the internal amount of this particular protein, CMP, was unaffected after the proteinase protection assay. So that shows you that that's actually an internal protein.
Sorry, Dr. Walt. Sorry to interrupt. I think we're one slide behind, perhaps, on what you're describing. I think if you use the green.
I don't think so. What I see on the screen is this field overall has struggled. Is that what you see?
Yes. On the lack of candidate validation or the poor immunocapture candidate selection?
Lack of candidate validation.
Okay. Yeah, if you can go to the next slide, I think then, where it shows your CNP protection assay data. Yeah, perfect. Yeah, thank you so much. Sorry, guys.
Okay, sorry. Yeah. Okay, so as I was describing, thank you, Ryan. Here's the non-treated. Here's the internal protein. You can see that those match up, and so this is actually after the proteinase protection. You can see that none of the CNP was actually removed from that process. So the opposite problem exists for the validation of internal targets. So GFP, as shown here on the right-hand side, has previously been used to define astrocytes EVs, but as we showed in the graph, it's predominantly secreted or extracellular because it's actually destroyed in the process, and thus it cannot be used to be validated as an internal marker for astrocyte EVs. I think that's showing correctly. Okay, so this is the workflow that we had planned to follow. As you can see on the upper left, what we do is we try to be very specific about the types of markers that we're looking for. The first thing is we want to make sure that these are cell specific. So for example, if we want neurons or astrocytes or oligodendrocytes, or microglia, sort of the four canonical types of brain cells, where we're trying to isolate EVs that are released from each of those specific types. First, we want to identify cell specificity. We want to make sure that they're brain specific, and then we want to make sure that they have a tetraspanin, that is, that they are transmembrane, because we're interested in pulling those proteins down specifically from CSF or from plasma. So it's the intersection of those this Venn diagram that comprises the proteins that check the boxes on all three of those criteria. So we are working through the steps that are outlined here, meaning what we want to do is first we want to run Simoa assays for each of the targets that we're interested in. We want to make sure that they elute in the EV fraction, that is, they're associated with the EV fraction and not the free protein fraction. We want to make sure, as you can see on the lower left, that they have both the isoform and topology that we want, meaning that they have the tetraspanin, that is that part of the protein that actually can insert into a protein, that is not a splice variant, that does not contain that region, and that it actually has the isotype that we're interested in. And then, of course, we're interested in developing the immuno capture protocols to be able to then pull those things down and isolate the EVs and then analyze the contents of those EVs. So we tried this approach, and every protein that we identified as a potential candidate turned out to fail. And so this is why we turned to Olink. So I know that this has been a long introduction, but I just wanted to explain the sort of challenges that we encountered with both pulling things down specifically and with all the previous literature that had demonstrated something that actually was not the case. So before I get into the specifics here, I want to mention that the work that I'm about to present was performed by a wonderful team of researchers here, led by Maya Norman, MD, PhD, who I was hoping would be able to accompany me today, but she's doing her clinical work today. So, and it was recently published in the Journal of Extracellular Vesicles. And the team consisted of Maya, of Adnan Shami Shah, postdoc, Sydney D'Amadio, Ben Travis, both RAs, Dima Ovanesian, another postdoc and staff member, and Tyler Dugan, a graduate student who just left the lab recently. So the challenge with looking at all these SEC fractions is that the early fractions have very little protein, and the late fractions have abundant protein. So they contain albumin and all sorts of lipoproteins like ApoB, and the late fractions therefore cannot be adequately analyzed by things like Western blot or mass spec, which are techniques that you'd think would be useful for discovering these kinds of markers. While Simoa works very well for this kind of analysis, and that's sort of the workhorse for my laboratory, we were developing these assays one at a time, and we have a bunch of different candidates that we were interested in exploring. So this was quite costly, both in terms of the time that it took to develop the assay and then to test it with these EV fractions, and also it was expensive. So we decided to run an Olink screen on fractions 6 through 15 of 4 healthy CSF samples, and we analyzed each of those fractions across the 5,420 proteins in the Olink panel. We overlap this data with the brain RNA sequence database to find proteins that were enriched in each cell type that we're interested in. Again, astrocytes, oligodendrocytes, neurons, microglia, etc. And then we overlaid those further with a platform called Deep TMHMN, which is a deep learning protein model-based algorithm that detects the topology and conformation of various proteins, particularly in the alpha and beta barrel families. So this protocol allowed us to identify targets that were both transmembrane and internal, that were cell specific and had distinct EV peaks in fractions 7 through 10, which is the EV fraction that I was describing before. So we analyzed our samples first with Simoa using size exclusion chromatography to fractionate the CSF. And this showed us the expected the expected peak for CD81, as you can see on the right-hand side at the top set of samples. So these are 4 distinct CSF samples from healthy individuals, healthy participants that were kind enough to donate their CSF for the study. And as you can see, the CSF fractions, CD81 shows the expected peak that it's associated with the EV fraction. Below, you can see that we could detect CD63, also a tetraspanin, in the EV fraction using the Olink panel. So this gave us a lot of confidence that the Olink panel was able to recapitulate what we would observe with a specific protein-specific immunoassay such as Simoa. So that was good. We also carried out what's called nanoparticle tracking analysis, which allows you to look at the particle size distribution. And as you can see, here's on the right-hand side, here's the CSF fractions 7 through 10. You can see that the size of the EVs is sort of between 200 and 75 nanometers, which is what we would expect to see for these EVs in these particular fractions. The red lines represent the standard error of the mean for 5 measurements per sample, and you can see that we get very tight standard errors, and so you can see that the EVs from this are clearly in fractions 7 through 10. The graph on the right-hand side, shows that there's a little bit of carryover of those EVs into the later fractions, but that's expected. So we also showed for several known EV proteins and contaminants of EV preparations that they elute in the expected fractions. And so this is just to show you that the EV contaminant proteins, as shown on the left-hand side, all come in mostly in the late fractions, which is shown in the dark bars over here on the right-hand side of the left-hand picture. And then the EV-associated proteins actually distribute both within the EVs, but also there's some free forms of these proteins as well. surprisingly, only 10% of the transmembrane proteins that we identified with Olink turn out to have a peak that associates with the EVs. The vast majority of those those transmembrane proteins, or the putative transmembrane proteins, had a significant protein content in the late fractions. And this points to the need for this type of validation before selecting immunocapture targets. And I think is really fundamentally the underlying reason why all previous attempts have failed, is that 9 out of 10 of these transmembrane proteins somehow must have isoforms that really are not inserted into the membrane itself. So using the Olink platform, we were able to identify several targets for each cell type. So you can see astrocytes, endothelial cells, microglia, oligodendrocytes, and neurons. We're currently in the process of following up each one of these leads by making Simoa assays for each one of those and validating the use of those specific assays to further validate the use of these transmembrane proteins to be able to immunocapture them and then to break them open and identify, using various methods, the internal proteins that are present in those immunocaptured EVs. When we do those immunocaptures, I just have to make sure I sort of tie this back to something I said earlier, is we use the protein protection assay. So once we pull these things down, we chew up all the external protein, including the antibodies that we use to capture those EVs. And then we put in detergent to gently lyse those EVs, release the contents of those EVs, and then put them into a mass spectrometer, typically to be able to identify the proteins, to be able to validate that those proteins actually correlate with what we expect from each of those specific cell types. So we're now in the process, as I just mentioned, of validating the hits that we discovered through this fantastic tool of using that Olink panel and validating them to use those individual proteins as immunocapture targets and then internally validating them. The Olink panel, as I mentioned, was a huge time saver for us, so that we didn't have to validate each of the candidate tetraspanins that we had identified using that original bioinformatics approach. And we'll probably use the panel again once we've been able to sort of identify which of those proteins we can use to immunocapture. Once we've immunocaptured them, then it makes sense, if we can get enough material, use the Olink panel again to identify which of those which proteins are present. Given the high protein concentrations in the late fractions, we could not have done this validation with sort of the obvious choice, which would have been mass spec, because it really looks at everything. And we tried that. We just kept getting albumin and ApoB. And so this really enabled us to, the Olink platform really was the ideal unbiased method for our particular application. So I'll stop there, thank the coworkers that I already mentioned, and happy to entertain questions.
Okay, wonderful. Thank you for that nice talk. Maybe as the questions are coming in, I'd like to highlight just a couple additional recent case studies. I just have 4 or 5 slides here because I thought with the increase in EV research that we're undergoing or experiencing right now, it'd be nice to highlight the utility of EVs derived from other sample matrices. So Dr. Walt did a great job at highlighting the use of EVs from CSF. I've got just 2 very brief cases here on how it's been leveraged. EVs have been leveraged from urine and plasma, and then following which I'd be happy to get into the Q&A session. We have a few minutes remaining still, so we're in good shape to go through a couple quick studies. So in one recent study that I'm highlighting here was using urinary EVs to help identify biomarkers that were specific for kidney fibrosis. So one of the challenges historically is that kidney fibrosis is difficult to detect and treat because of the lack of really good biomarkers. And so this study took kidney transplant patients that experienced fibrosis, as well as healthy controls and non-fibrotic patients, isolated using a series of methodologies, including size exclusion, chromatography, as we just heard about, and nanoparticle tracking, as we also just heard about, and then ran those urinary EVs using one of our middle plex panels called Olink Target 96, which is 92 proteins run at the same time. So whereas Dr. Walt ran 5,400 proteins, this particular study focused on the use of 96 proteins. And as you can see, at a global level, out of the 92 proteins, 33 of those proteins were differentially expressed across patients with kidney fibrosis versus those that didn't. On the right-hand panel, right-hand side of the panel, you can see a substantial upregulation in this heat map of these 33 proteins, whereas to the left of center, you have a bit of, I guess, an intermediate phenotype where some proteins are upregulated, some are down, and those are the intermediate fibrotic individuals. And then on the far left are the non-fibrotic individuals, indicating that there are some global changes occurring among these proteins or the expressions thereof that would suggest potential biomarkers to distinguish fibrotic individuals from non-fibrosis, kidney fibrosis, that is. And I'm going to abbreviate most of the paper. If you're interested in this, there's a lot of interesting information in this paper. I'm going to focus on one specific aspect, which is a new potential biomarker of kidney fibrosis that they identified, which was MCP1. And of the 33 differentially expressed proteins, MCP1 was the most upregulated of those proteins in fibrotic individuals versus healthy controls in non-fibrotic patients. And you can see the Olink data in the top right, which they then went on to validate using an MCP1 specific ELISA, replicating the data in the bottom left panel that shows non-fibrosis versus fibrosis, and then took this one step further to bin those individuals into individuals with 0% fibrosis, 1 to 10, or greater than 10, all showing that using MCP1 alone, you could distinguish between those 3 different groups. And then finally, in the bottom right-hand panel is a rock plot showing the ability of MCP1 by itself as a single biomarker to distinguish fibrosis individuals from non-fibrotic individuals with an AUC of just over 0.8. And so MCP1 from this study of urinary EVs has been identified as a potential biomarker of kidney fibrosis and was orthogonally validated using ELISAs and may offer new insights into the complex mechanisms of kidney fibrosis and perhaps by extension into chronic kidney disease patients. And just as a final study, this one was interesting on 2 fronts. One was because it was isolating EVs from plasma, which is interesting for the reasons Dr. Walt mentioned previously, which is that it's a non-invasive sample type to obtain. But in this particular case, it was interesting for 2 reasons. One was because from an engineering perspective, these authors developed a new microfluidic device to isolate EVs. And then on the biological side, it was interesting in that they were looking at individuals with long COVID. So since COVID, individuals with long COVID have progressively been reporting increased cognitive decline. And a lot of the proteins that are dysregulated in those individuals overlap with the proteins that are also upregulated in Alzheimer's disease patients. And so what this study did is it took long COVID individuals with and without cognitive decline and compared their proteomic profiles to AD patients and HIV patients that similarly did or did not have cognitive decline as well. And they developed this EV microfluidic affinity purification technique that I'm showing in the middle here, whereby total EVs were precipitated initially from plasma using an exoquick reagent and then loaded onto a microfluidic chip that was decorated with monoclonal antibodies that we heard Dr. Walt refer to previously. And then those captured EVs were then photoreleased into phosphate buffered saline. So an interesting bit of engineering to isolate neuronal enriched EVs from plasma to then run using Olink Explore, our neurological panel, which is approximately 380 proteins in a single run with our NGS readout. And what they found was among the long COVID individuals that did or did not have cognitive decline, they identified 13 specific differentially expressed proteins. And when taking the best performing protein in isolation, as you can see in the rock plot on the right, MESD was able to distinguish long COVID individuals with and without cognitive decline with an AUC of nearly 0.8. Similarly, in the AD patients, 14 differentially expressed proteins identified, but selecting only the single best performing marker on its own was able to distinguish those individuals with AD versus healthy controls with an AUC of about 0.9. And then finally, among the HIV patients that did or did not have cognitive decline, 41 differentially expressed proteins identified from this panel of 380-ish proteins. And when looking at only the top performing single biomarker, CPA2, identified an AUC of approximately 0.8 again. So this particular study was interesting, as I mentioned before, because it isolated EVs from plasma that were neuronal enriched to begin to tease apart the differences between the proteomic profiles of individuals with and without cognitive decline, either in long COVID with HIV or AD. Also interesting is, in addition to distinguishing between these groups, they performed these network analyses shown on the right that were also able to lend insights into the mechanisms or similar mechanisms that might occur between these different disease states. And they noted in this paper that some studies such as this weren't previously as informative because they lacked the ability to test as many proteins in one run as was capable using our 380-plex panel. Which goes back to what Dr. Walt referred to a moment ago, which is that you can use these panels to screen a large number of proteins in tandem to vastly increase your throughput. So in conclusion, as we've seen a few times today, PEA is compatible with a wide variety of sample types and including EVs, which is an increasing a field with increasing research interest. The sensitivity of PEA and the requirement for low sample volumes makes it quite advantageous for studies such as this when using EVs or microvesicles or things of that nature that are precious or invasive or difficult otherwise to obtain. And Olink remains the only fully scalable technology that uses the exact same capture reagent from end to end, enabling seamless integration from your discovery-based studies all the way through to clinical implementation. And so in lieu of a thank you slide with the questions here, I just wanted to first thank you all for attending. We appreciate your attendance today, but I also wanted to mention that we're excited to return on October 8th of this year with our next iteration of Olink Proteomics World. We'll have over 20 leading speakers across various key research areas for a day of insights, innovation, and of course, inspiration. We'll have the day broadcast across 2 different time zones. There are 3 dynamic tracks that we will have. So multi-omics in precision health, population proteomics, and disease-focused applications. There are also expert-led tutorials, panel discussions, networking events, and of course, virtual booths and posters. So I'll leave this slide up during the Q&A session, but if you'd like to scan the QR code and please register for Olink Proteomics World, it's coming up on October 8th. And with that, I think we can go to a few questions. Let me open them here. And with us today, we also have Olink's Senior Field Application Scientist, Joe Crusoe, who will help with any of the technical questions that you might have as they come in. So just go do a quick perusal, go to the bottom. There's quite a few here that we have. So we'll get through as many as we can, and then we'll try to follow up with the remaining ones as we go. So maybe the first question to Dr. Walt, how do you envision the CSF-EV protein dataset being used by the broader research community? So what steps are needed to move it forward in its application of, for example, neurological diagnostics?
Yeah, so it's a great question. That we published the paper, as I mentioned, in Journal of Extracellular Vesicles. The set of proteins that we identified is now publicly available, and the community is free to go explore those initial hits as they see fit, using sort of their favorite approach. Again, I think they just have to be careful, particularly with with EVs, to make sure that they're following the proper protocols to ensure that what they're looking at is actually what they think they're looking at, not only by making sure that the antibodies that they're using, for example, to perhaps pull something down, are specific. For example, I've gone through some of the questions. One of the things that people have to be careful about is, if you're using an antibody that is specific to an intracellular loop of the protein of interest, you're not going to pull down an EV. You have to make sure that it is recognizing the extracellular, or the the outside of the membrane, the external epitope of that particular candidate protein. And so we've seen this many times, where in the literature, people will pull something down with a particular antibody, and it's just not specific for the particular epitope that actually reflects that external protein. So to answer the question, this is sort of a free-for-all at this point. Anybody's welcome to look at this set and explore the proteins that are of particular interest to them.
Wonderful. Yes, it's certainly a nice resource to have initiated anyway. Maybe the next question for Joe, it's a bit technical in nature. And when working with precious samples, or samples that are only available in limited volumes, is there a need to run replicates on all of these panels?
Yeah, that's a very good question also. So like you mentioned earlier, one of the great aspects of Olink technology is that you do require very small starting volumes to actually run the assay. So maybe 1 microliter for midplex, or about 2 microliters if you're running our Explore HT platform. So that really helps with specific matrices like EVs, which don't really typically produce a large volumes. And then another aspect is that we don't really require technical replicates of your samples either. So this is due to inclusion of an internal standard when we're calculating our MPX values. And then we just find in general that the biological variability supersedes the technical variability in the assay. So you wouldn't really require technical replicates of your samples as well. Having said that, in some cases, we don't really require biological replicates for biofluids, but in some cases with alternative matrices, when you're running a little bit more sample prep involved, such as EV preparation, then in that case, then maybe biological replicates might be recommended as well. But I think for a lot of these EV studies, I think typically they're just normally run them as a single kit in general, I would say.
Okay, great. And going back to Dr. Walt, could you expand a little bit on your decision to remove the EV corona for biomarker discovery? Is it foreseeable that those proteins would also be informative?
Yeah, it's a really good question. Yeah, the reason that we end up doing that protease protection assay, which as I described, chews up all the external proteins, is that we have found multiple proteins. L1CAM is a great example that just stick non-specifically. So there's non-specific binding to the EV membrane, and others have reported long ago, 10 years ago, reported that for example, they were isolating L1CAM pull down EVs, and they could detect alpha-synuclein in Parkinson's disease patients that were in the EVs. It turned out that that alpha-synuclein, one of the key proteins in Parkinson's pathology, also stick non-specifically to EVs. So these, the alpha-synuclein turned out to be, correlated basically with the plasma concentration of alpha-synuclein, and it had nothing to do with specific pull down of a brain-derived EVs using L1CAM. And so even though we'd love to be able to isolate those proteins, the reality is that if you don't get rid of the protein corona, you end up with a lot of proteins that just are non-specifically stuck. The way that we get the identity of those tetraspanins is if you use mass spec, you can actually detect the residual peptides that are present, and both present within the membrane, but also the intraluminal loop of those proteins. And so you can still get specific proteins that are associated with the EV, but you've chewed away the extracellular components of those proteins.
Okay. Wonderful. I'm keeping mindful of time. The questions that are in the inbox, we'll do our best to answer them after and follow up with individuals. So there's a couple other comments that I can just make a few statements about. The one is regarding recording. That is recorded and will be available on demand. And we do have protocol suggestions and guidance when prepping CSF and EVs. This question came up a couple times, but we don't have time to present it or post it to Joe. However, if you do reach out to us and you do want suggestions or protocols for how to isolate various different sample matrices, we do have guidelines that we can share with you on those topics. Just in the last minute here, I'm going to advance to a couple series of poll questions that we would appreciate if you could fill out. And we use this to collect information on the platform and people's interest, and it helps guide future talks as well. So if you have a moment that you can still take to fill out these survey questions, we would greatly appreciate it. And as I mentioned a moment ago, we will have this available on demand after. So if you couldn't attend all of it, you can catch it then. And while I'm doing this, I would just like to thank Dr. Walt again. It's been my pleasure to work with you to have this webinar come to life. I appreciate your time and all of your effort in helping us do this today. So we greatly appreciate that.
Yeah, and I thank the Olink team and the LabRoots team.
Wonderful. Okay. With that, I can hand it back to Pernilla.
Perfect. I will go ahead and close this out from here. Thanks, Ryan. And thank you, gentlemen, for your time today and for your very important research. We would also like to thank LabRoots and our sponsor, Olink Proteomics, for underwriting today's educational webinar. Before we go, I would like to thank the audience for joining us today and for their very interesting questions. Questions that we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. As a reminder, this webinar can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. All right, until next time, take care, everybody. Goodbye.
Thank you.